Over to you, Ashutosh.
Good afternoon, everyone. Am I audible, Divya?
Yes, you are.
Okay. Good afternoon, everyone. Very warm welcome to all of you in yet another chapter of our webinar series, Let's Talk Genomics with Experts. The theme of today's event is interpretation of variant detection in NGS. NGS has revolutionized the field of molecular diagnostics and it generates large volume of genomic data. These sequencing data from human genome can be used for developing an understanding of diseases along with refining its diagnosis, individual clinical care, predicting the drug effects, diagnosis of infections, tracking epidemics, and many more applications. To understand more on these aspects in today's discussions, Dr. Dalal will talk about some of these applications of NGS in diagnosis of genetic diseases and principles of interpretation of variant detection. So to introduce Dr. Ashwin Dalal, he heads the diagnostic division at CDFD Hyderabad, and he has been instrumental in establishment of Department of Medical Genetics at Nizam's Institute of Medical Sciences, Hyderabad. He is also adjunct visiting professor in the Department of Medical Genetics, Kasturba Medical College, Manipal. He is a founding member and present president of the Society for Indian Academy of Medical Genetics. He has more than 130 papers to his credit, published in national and international journals. His research interest includes the identification of novel genes for single gene disorders and molecular analysis of genetic diseases. He has contributed chapters in textbooks for MBBS and MD students and has interest in teaching medical genetics in medical colleges and conferences. Dr. Dalal has been a member of various task forces like DBT, ICMR, and DST. And in his current role at CDFT, his lab offers comprehensive diagnosis, genetic counseling, and prenatal diagnosis of genetic diseases. So with this, I would like to call upon Dr. Dalal to take today's sessions. But before that, as I can see, we have a great mix of students, lab personnel, faculties, clinicians, and others in the audience today. So please feel free to interact with us and Dr. Dalal in question answer session. And also while sessions are going on, if you have any questions, please put it in chat box. So with this, Dr. Dalal, we are eager to hear you now. What do you say?
Hello. Yeah. Thank you. Thank you, Ashutosh. Thank you, Premas, for inviting me. And today we'll discuss about this very common as well as important topic on interpretation of variants detected in the next generation sequencing. So as we all know, next generation sequencing has really become a routine diagnostics available at multiple places throughout the country, and there are different types of next generation sequencing methods available, platforms available. However, everything ultimately boils down to the variant that is identified, and that is the most difficult part now, because we don't have an instrument or a software which can tell us that this variant is disease causing or not. And that is where the input from the clinician, the input from the various databases help us to make that ultimate decision whether this is a disease causing variant or not. So today we'll try and discuss about how next generation sequencing can really help in the clinic for identification of variants, as well as once a variant is identified, how we are going about deciding whether this is a disease causing variant or not. So let us start with the basic thing, that what is genetic testing. So genetic testing we can define as analysis of human DNA in its various forms. So it could either be in the form of chromosomes, where we call it as the cytogenetic testing. At the level of DNA, we will call it as the molecular genetic testing. And at the level of protein, we call it as the biochemical genetics, where we study either enzymes or biomarkers. Now, related to DNA based testing, all of DNA based testing that is available or that we do in the lab is basically based on these two principles, that is complementary hybridization and polymerase chain reaction. If we understand these two principles, then we can understand all types of genetic testings that are available. So the complementary hybridization principle tells us that G always pairs with C and A always pairs with T. And this kind of pairing is made use of in various genetic tests like dot blot, reverse dot blot, up to the most recent microarray technology also is based on this principle. The next principle is the polymerase chain reaction, where we use this method to amplify the amount of DNA available so that we are able to visualize it in some or the other form. And this principle is the basis for all kinds of tests ranging from a simple PCR based test to PCR RFLP, ARMS PCR, next Sanger sequencing, and up to the next generation sequencing. So all these tests are based on basic these two principles. The next important thing to understand is what are we looking for using these tests. So when we want to do a sequencing, what we are basically looking for are the mutations. So in humans, we have mainly these three types of mutations. Either it could be a point mutation, or a triplet repeat mutation, or a large deletion duplication. So point mutations are mutations found commonly in diseases like thalassemia or cystic fibrosis, where these mutations are small mutations distributed throughout the gene, which could be either single nucleotide insertion, deletion, or sequence or nonsense mutation. Triplet repeat disorders are diseases like Huntington disease and spinocerebellar ataxias, where there is increase in the size of this triplet repeats, which leads to the disease. And you have deletions and duplications in diseases like Duchenne muscular dystrophy or spinal muscular atrophy, where large deletions and duplications lead to the disease. Now, the problem is that we still do not have one test which can detect all these. But as a clinician or as a laboratory scientist, you would be interested in a test which can detect all mutations in one test. So now at present, the gold standard for detecting point mutation will be Sanger sequencing. The test of choice for triplet repeat disorder will be a genotyping using a small PCR or a triplet prime PCR. And for large deletion duplications, you would either use MLPA, real-time PCR, or a array comparative genomic hybridization. So we still await a test which we feel whole genome sequencing with complete analysis would be a test which can detect all these. But as of now, we have to use these different kinds of tests to identify different types of mutations. Now, if you look at Sanger sequencing, what's the principle of Sanger sequencing is that we decide which part of the gene that we want to sequence. We do a PCR using flanking primers, take the PCR product and do a second round of PCR using a single primer and run it in a capillary electrophoresis to get this nice peaks of A, T, G, and C. However, Sanger sequencing, like any other investigation, has its own problems. It is difficult to use when we do not know which disease we are dealing with or which gene we want to sequence. It is difficult to sequence large number of exons. For example, in osteogenesis imperfecta, you have two genes in type 1 which have almost 101 exons, or in patients affected by diseases which are caused due to multiple genes. Again, these phenotypes, it becomes difficult to do it by Sanger sequencing. So prior knowledge of genome is very important in Sanger sequencing, which part you want to sequence. At a time, you can sequence only limited amount of sequencing in a Sanger sequencing, and it becomes costly and labor intensive for large sequencing. And that is where the next generation sequencing came into picture, which is a massively parallel sequencing, where again you start with the genomic DNA, but here you fragment this DNA into small pieces, adapter ligate them, do a few rounds of PCR, and then ultimately sequence everything that is available. And that is where the bioinformatic analysis comes into picture, where it helps us to identify the variant. And as we all know, the cost of sequencing has dropped such drastically that today you can sequence a whole genome for about 50,000-60,000 rupees, whereas it took $2.7 billion to sequence the first human genome. So because of this drastic decrease in cost, we have next generation sequencing being routinely used in the clinic. However, NGS has its own problems because of millions of sequencing reads and You need to map it to a proper place in the genome, and then detecting the variant is a very hard task. So it is not a machine, it's not a human readable. Like in Sanger sequencing, you get this nice peaks of ATGC, which any human can look at it and decide whether mutation is present or absent. Whereas the data which comes out of NGS analysis is not really human readable, and that's why you need a good bioinformatic support to interpret these results. So in the clinic, we have different approaches through which we can use next generation sequencing for identification of variants. Either it could be a targeted resequencing or a exome sequencing or a whole genome sequencing. Targeted resequencing refers to sequencing of a group of genes which are responsible for a phenotype. So this could be a phenotype like cardiomyopathy or a deafness or blindness, where we know almost more than 70-80 genes are known to cause this phenotype. So you can group them together and sequence them together. Or you could have other panels like an inherited diseases panel, a cancer panel, or a clinical exome panel. So this is an example of a familial cancer panel, which you can see multiple genes which are associated with that particular phenotype are included there. So the clinical utility of a panel is sometimes when you want to sequence a large gene, like if you want to sequence a gene like Duchenne muscular dystrophy, which is a very large gene, there you can use a panel to sequence that large gene. Or if you want to see, if you do not know which gene you are dealing with, like in a deafness or blindness, there you will use, or sequencing of group of genes for a similar phenotype. So you could have these kind of phenotypes where you want to sequence group of genes, and then always we want more from what we do, and that is how this clinical exome panel was born, where you have a panel of about 5,500 to 6,000 genes which are known to cause any human disease. So human genome consists of about 20,000 genes, but we still do not know the diseases caused by almost 15,000 genes. So we have a clinical exome panel, or it is also called as a focused exome or a Mendeliome, where we sequence all the genes which are known to cause any human disease, and that is very useful in a clinical scenario. However, slowly the cost of doing a clinical exome panel and a whole exome sequencing is almost becoming similar, and hence nowadays mostly what we prefer to do is a whole exome sequencing and then analyze the variants. Now, what about whole exome versus whole genome? So exome is the coding part of the genome. So it consists of only 1% of the human genome. But if you look at the cost, it is a lot more easier to sequence an exome compared to a whole genome. And if you look at the number of variants, you get about 22,000 variants in an exome, as opposed to 23 lakh variants in a genome. So it's always easy to find 1 in 20,000 compared to 1 in 20 lakh. And that is why in clinic, what test is preferred as of now is a whole exome sequencing. However, hopefully in the near future, as the whole genome sequencing becomes more easier to analyze as well as more affordable, ultimately I hope that in the next 3, 4, 5 years, we all would be doing a whole genome sequencing and then analyze the genome based on our requirement. So exome sequencing has clinical utility mainly for screening mutations in unknown phenotype where we do not know what's the exact gene involved. And in research, it has helped us a lot in identification of novel genes for various Mendelian phenotypes. Now, finding a variant in a exome is almost similar to finding a needle in a haystack. And here, this kind of strategy is not really going to help us. So what we would like to do is that we gather the hay together, have small bunches of that hay, one bunch at a time, and we find lots of needles there. We use some sort of a sieve and then reach our ultimate needle. And this is the same thing that a bioinformatic pipeline would do for us, where we begin with the genome and the raw data is the FASTQ file, which maps to the reference genome. We get a BAM file, and through the BAM file, we identify the variants. That is the VCF file. And when this VCF file is annotated and filtered, that is when we get our single nucleotide variant or the deletion duplication that we are looking for. So this complicated bioinformatic pipeline is really going to be helpful so that the machine readable data can ultimately help us to identify that particular variant. And that is why it is very important for today's clinician or today's life scientist to be well versed with the computational approaches, because all these large data that are generated, either it is transcriptome, exome, genome, all these are not human analyzable, and that is why very important it is that we have some knowledge of the computational setup. So let's see this one example where how we go about using exome sequencing to identify a variant in a clinical situation. So this is a 6-year-old boy who came to us, and he was overweight from birth. So he was 4.2 kg when delivered. He had prolonged neonatal jaundice and documented hypoglycemia in the neonatal period. And at the time of examination, he had increased liver size and kidney size. So it is hepatomegaly, renomegaly. And there was generalized overgrowth with all head circumference, weight, and height above the 95th centile. So all these clinical features were suggestive of overgrowth syndrome, most probably a Beckwith-Wiedemann syndrome. So this is our clinical diagnosis that we are going with. Now this clinical diagnosis, once we have to find out what is the mechanism of disease in this particular condition. So the mechanism of disease is known to in Beckwith-Wiedemann syndrome are large number of mechanisms. So it could be caused due to a chromosomal translocation. So that's why we did a karyotype and that was normal. It is known to be caused due to deletions in certain genes. So array CGH was done, which was negative again. It is known to be caused due to methylation problem at the IC1 locus, that was again normal. And then it is also known to be caused due to point mutations in this particular gene CDKN1C, and that was also sequenced and found to be normal. So now once we have ruled out all mechanisms of Beckwith-Wiedemann syndrome, that is when we thought we should take up this patient for a whole exome sequencing, because the known mechanisms are not yielding any result. So whole exome sequencing, this is how you get the variant, and then we go about filtering using different strategies, which we will discuss soon. And using these filtering strategies, ultimately we reach to this one particular gene, that is the GPC3 gene. Now this GPC3 gene, there was a deletion of single nucleotide T at this particular position, which leads to a frame shift mutation, and this is present on the X chromosome. So it is a hemizygous, not present in databases with a high CAD score. So it was interpreted as a disease causing variant, and this is how we look at that particular variant in a next generation sequencing. So instead of the peaks, what we get is all these next generation sequencing reads, and we can see here this is the homozygous or the hemizygous deletion of this particular nucleotide here. And the same always we go and confirm using Sanger sequencing. So you can see that there are 2 T's in the normal person, and this here is a homozygous deletion, only 1 T is present, and the mother was carrier for the same deletion. So ultimately at the end of it, we have the final diagnosis in this patient. This patient is a case of Simpson-Golabi-Behmel syndrome due to pathogenic variant in this particular gene, and this phenotype is very similar to Beckwith-Wiedemann syndrome. So that's why it's important that we follow the genotype as well as the phenotype. So once something is identified in the gene, we go back and see whether it matches with the particular phenotype or not. Now this is another situation where we have this patient who had global developmental delay. He has congenital cataract, deep set eyes, as you can see here, small hands with very small fingers, and this large epiphysis here. The metaphyses are large, and so you have a skeletal dysplasia with developmental delay with cataract. So this was a multi-system involvement. So we were sure that we are going to get some or the other mutation. So since the clinical phenotype was not fitting in a known syndrome, we went ahead and did karyotype, microarray, exome sequencing, all was found to be normal. So what would be the reasons of a normal result? One is all exons may not be captured or sequenced. So exome sequencing, we should always remember that exons are captured in this. So some exons may not be captured, or they may not be sequenced, or sometimes the mutation may be in the intron or non-coding region, which again will not be detected in an exome, or the mechanism of mutation may be different, like a triplet repeat expansion or a chromosomal. Hence, Normal exome report does not mean that the disease etiology is non-genetic, and we should go ahead with further testing based on the phenotype, like whether we are suspecting a intonic mutation or a triplet expansion or a methylation analysis. It will depend on the phenotype that you can investigate further. So every investigation has its own advantages and disadvantages, and you should keep that in mind while ordering a test or interpreting a result. So now that was about exome sequencing and how we use it in clinic and how it helps us. Let us go on to now about how we are going to interpret a variant. So whenever a variant is encountered in practice, this is true for a life scientist who is analyzing the exome, or it is true for a clinician who is sitting in the clinic and he gets a report from the laboratory and he wants to do variant interpretation himself. So why do we need to interpret a variant? So that is an important question that we need to deal with first. Why do we need to interpret? So what we can see in this picture here is that at the first glance, it looks like these are only some dry leaves around here. But if you look closely, then what we can see is that it is actually a lizard which is masquerading as the dry leaves. Okay. So what looks very harmless can turn out to be harmful. On the other hand, what you can see here is that this is a very ferocious kind of animal, so a very harmful kind of animal, but the human is very close to this animal, so this harmful animal has become harmless to this human. So the same is with variants. Harmless looking variants can sometimes be disease causing, and the classical example is this Hutchinson progeria, which is a very severe disease of aging, and it is caused due to a synonymous mutation which mostly we will disregard. On the other hand, a harmful looking variant like a stop codon mutation can sometimes be a normal polymorphism, and hence it is very important that we should try and interpret each variant in a proper way so that we don't make any mistake, and that will percolate into problem for the patient. So let us understand some of the words that we use while interpreting a variant. So variation is difference in DNA among different individuals as compared to the reference genome. Mutation is a word used for heritable alteration or change in the genetic material, and polymorphism is defined as inheritance of a trait controlled by a genetic locus with two alleles, in which the least common allele has a frequency of 1% or more in the population. Now, as a practice at present, we do not like to use the word mutation, and the word that is used is a variation. So we could either call it as a benign variation or a pathogenic variation, and hence we will not be using the term mutation. So now let us see, can I do a variant interpretation? This is a very important question that comes to mind of many people, that do you need a lot of education or a lot of practice to do a variant interpretation? Now variant interpretation question can come to a computational person or a life scientist who is sitting in a lab and trying to identify a variant, or it can come to a clinician who is trying to interpret. But what I feel over the years that I have seen myself as well as my friends doing the variant interpretations is that anybody can do a variant interpretation. And today I hope that we will try and see, if we follow the particular steps in a stepwise manner, then anybody should be able to do a variant interpretation. And these are the mandatory steps or the important steps. If you go through each of these steps one by one, then I'm sure that you should be able to interpret any variant that you are looking for. So the important steps include, first, we will try and find information about the location and functional effect of that particular variant. Then we would like to know what is the frequency of that variant in normal population. Then we want to know whether this variant has been reported in patients with similar phenotype earlier. Then we would like to predict this variant effect using various software or tools. Then we would like to do a Mendelian segregation analysis followed by a functional assay. So when all these steps are done, then any variant that we find can be put into one of the classifications that are used. So let us first go on to see what is the importance of this location and functional effect of the variant. If you look at the structure of a gene, so the structure of a gene consists of a promoter followed by the 5' untranslated region, and then you have the start codon ATG followed by the first exon, then an intron, exon, intron, exon, and then you have the stop codon here followed by a 3' untranslated region. So this is what is the structure of a gene, and at the intron exon junction you have this consensus sequences called as GT, which is the donor, and AG, which is the receptor. So once you have this information available with you, then you can do a location based as well as a functional classification, and that can give you some idea regarding what could be the property of that particular variant. So location based classification can be that the variant could be either in the exon or the intron or at the splice site or in the 3' untranslated region, the 5' untranslated region, the promoter or the enhancer, or in the non-coding region. Now, as a general practice, what we will feel is that exonic variants and splice site variants are the variants which are likely to be disease causing. Intronic and variants in the regulatory regions are less likely to be disease causing, although they can also cause disease at some positions. But overall, you would first like to look at the exonic and the splice site variants. Similarly, based on functional effect, you can classify them as missense, nonsense, frame shift, in-frame or silent variants, or also as synonymous and non-synonymous variants. So let us see each type of variant and how that information can help us to make some judgment about that particular variant. So first is missense variant can result in disease either through alteration of the protein function. So here you can see this patient is a 5-year-old patient who came with 4 species and the typical dysostosis multiplex to us. So we have a clinical diagnosis of mucopolysaccharidosis and his enzyme assay for aryl B was low. So we have a diagnosis of MPS type 6. So we go and sequence that particular gene and we find that there is a homozygous mutation in this particular gene. So this homozygous mutation now is not known disease causing variant, so it's a novel variant. So you would like to use some sort of a in silico method, and you see that this variant is present at the catalytic site of ARSP protein, and it forms coordinate bonds with the calcium ion, which tells you that this missense variant is likely to be disease causing since it is present in the catalytic site of that particular enzyme. So missense variant can lead to alteration of protein function. A nonsense variant can be either within 55 bases of the next exon junction or beyond the 55 bases, based on which you will think that whether nonsense mediated decay will happen or not. So nonsense mutation can lead to disease either by formation of a small protein or by total absence of the RNA because of a nonsense mediated decay. So this was a patient that we had who had global developmental delay, spasticity, and his exome sequencing identified this particular stop codon mutation in AIMT2 gene. So you can see now this is a nonsense variation, and then you could check for the nonsense mediated decay, and here we could see that there was a partial nonsense mediated decay happening because of this stop codon. So nonsense mutation can lead to disease based on a small protein or nonsense mediated decay. A splice site variation can lead to disease either by inclusion of few intronic bases into the exon or by exon skipping. So you have this, we have this particular patient who had this classical clinical phenotype of Schwartz-Jampel syndrome, and when we did exome sequencing, we found a mutation at +5 position inside the intron, and this was predicted as a splice site variation. So once it is predicted as splice site variation, we took the RNA and made cDNA from this patient, and you can see that the cDNA is showing a smaller base pair product, and the cDNA sequencing showed that because of this mutation there is skipping of this particular exon, and hence deletion of all the nucleotide, the protein, the amino acids coded by this particular exon. Similarly, a frame shift variation which results from a deletion duplication can lead to either a premature termination codon, which again can undergo nonsense mediated decay, or a small protein, or it can alter the amino acid sequence beyond that particular frame shift mutation. So frame shift mutations also can lead to disease based on this. So you can see this particular patient here, we have two females affected with leukodystrophy kind of a disease, and exome sequencing here identified a single base deletion in this particular gene. So single base deletion here, you can see the homozygous deletion. We can say that what can this lead to? So we write all the nucleotides in a line, and then we change the frame based on the particular deletion. And when we change the frame, we see that there is a stop codon coming only 23 amino acids after that particular mutation. So it is going to lead to either a smaller protein or a nonsense mediated decay. So that is a way in which you can identify a particular type of mutation either based on the location or the functional effect, and then try and think what this variant could result into. So that information you will be taking into consideration while making the decision that it is a pathogenic variation or not. The next evidence that you are going to collect is the frequency of this variant in normal population. So why this is important is because if you look at all humans throughout the world, then 99.9% of the genome is similar in all humans. It is just this 0.1% difference that makes us all different from each other. So this 0.1% difference in different people is something what we call as the polymorphisms. Now this polymorphism frequency is different in different populations depending on the ethnic origin of that particular population. So it is very important that when we look at the variant, we have to know the polymorphism frequency in that particular population to say that whether it is a polymorphic variant or a disease causing variant. So it is very important, that's why, to have population databases of each of the ethnic populations throughout the world. And this is a very important chart which we need to keep in our mind about the effect size of the variant and the frequency of the variant. So you could have certain variants which are rare in the population. So they are very rare in the population and they have high effect size. These are the ones which are causing all the single gene disorders. On the other hand, you could have rare variants which have small effect, and these are the variants which are likely to be involved in either digenic inheritance or oligogenic inheritance. Then you can have, on the other hand, common variants with mild effect, and these are the variants which are going to be involved in the causation of multifactorial disorders, and mostly these are studied by the genome-wide association studies. So what in the exome sequencing of rare disorders, we are mainly bothered about these variants which have high effect size and are rare in the population. So the filter that we will use for identifying whether a variant is disease causing or not is that we try to see if a variant is less than 1% in the population, then it is likely to be disease causing. However, this may not always be true, and we need to keep in mind about certain variants which are disease causing, but they are more common in the population. Just like thalassemia is more common, so you will have higher frequency of thalassemia variants in Indian population. Similarly, for Caucasian population, you could have cystic fibrosis mutation which could be higher in that population. But as a general rule, you would like to see any variant which is less than 0.01 frequency to be likely to be disease causing. So for doing that, we have different databases that have been developed over the years. So first one was the 1000 genome database, where we have a large number of genome sequence. Then you have the exome variant server. Then you have the exact, or you have the genome AD, which is a recent version of genome AD. And then you have this complete genomics, or the great Middle East variome. Then we have the Genome Asia project, which was again a large project, and then there are others. So these are large consortium projects which have made available all these population variants which are made use of while interpreting the population frequency. So each of these databases have their own websites which you can visit, but otherwise mostly we try to incorporate them into the variant filtering Excel sheet. So if you happen to identify a particular variant, you can look at that variant frequency in all these different population databases. Similarly, from the Indian population, we still don't have a large database. There have been some small databases which have been published consisting of few individuals, and we were happy to publish one of the database last year of about 1500 exomes, and the same is available on the website of CDFD, and anybody can freely download this frequencies in the Indian population. Although it's not a very big database, but at least it is something that we can make use of for the Indian population. And now a major database that is coming up is the Genome India database, where about 10,000 Indian population is being sequenced, and we hopefully should get this database up and running in the next 1 year or so. So that will be very helpful because we come across many of the variants which are very rare in the Caucasian population, but they might be very common in the Indian population, and hence we need to analyze very few variants if we use the Indian databases to filter them. The next evidence that we are going to gather is that is this variant reported in cases with similar phenotype. And here you have different databases like either ClinVar, OMIM or PubMed, which are free databases, and the Human Gene Mutation Database is a paid database. Multiple such paid databases are also available, which help us to know whether same variant has been reported earlier in a patient with similar phenotype. If we find such a report, then it is very easy. We can directly report that this variant is a disease causing variant. However, if we find that this variant has not been reported earlier, so it's a novel variant now, and this novel variant now will use various prediction methods to predict whether this variant is likely to be disease causing or not. Now, most of these prediction tools, they make use of different properties of the amino acids or the sequence. So it could either be the conservation score for the evolutionary conservation of the particular amino acid, or it can be the sequence properties, the structure of the protein, or various other annotations. So example, if you look at the conservation across species, you can see that this amino acid is conserved across human to cow to rat to mouse. So any mutation in this amino acid is likely to be disease causing. Whereas if you look at this amino acid, it is not same in all species, and hence a mutation at this particular amino acid may not be disease causing. So this is one type of evidence that we can use. Secondly, we look at the biochemical properties. So if a hydrophobic amino acid is changing to a hydrophilic amino acid, or a charged amino acid is changing to a negatively charged is changing to a positively charged, or a polar is changing to non-polar. So this kind of information can be used to again predict whether it is likely to be damaging or not. Or you can look at the 3D structure or the 2D structure of the protein and see whether this structure is likely to be affected because of the change of that particular amino acid. So taking all into consideration, various tools are available like SIFT, Polyfen, Mutation Tester, Mutation Assessor. A large number of these tools are available, more than 100 are there, which can help us to make a prediction that whether this variant is likely to be disease causing or not. So each of these tools has their own website, like the Human Splice Finder is for the splice sites, and then CAD is a very important tool which is used by a lot of people because it gives a score. So using the CAD score of more than 20, again you can try to interpret the variant. After this, the very important step is Mendelian segregation in the family, because we are dealing with Mendelian disorders. Once a variant is identified in exome sequencing, you would like to know whether it is segregating in Mendelian fashion expected for that particular phenotype. So you may get a heterozygous variant or a homozygous variant. So if you get a heterozygous variant in the patient, then you can check the parents, and when you check the parents, if both parents are normal for that particular variant, then you will say that this variant is a de novo variant and hence more likely to be pathogenic. On the other hand, if one of the variant is carrier of this particular variant and that parent is asymptomatic, then you will feel that this is less likely to be pathogenic. However, we need to keep in mind about the possibility of incomplete penetrance. Similarly, if you find a homozygous variant, then the possibility in the parents is that both parents might be carrier for that variant, and this follows the typical autosomal recessive kind of Mendelian segregation, and hence you will say that maybe this variant is likely to be pathogenic. However, if one of the parent is homozygous for the same variant and the parent is not affected, then you will feel that this variant is unlikely to be pathogenic because this parent is not affected. Why this proband would be affected? And sometimes you may get that one parent is a carrier, the other is not a carrier, and there you will have to keep in mind other mechanisms like uniparental trisomy or a large deletion or disputed paternity needs to be kept in mind. So Mendelian segregation analysis is a very important step to be followed in analysis of the variant to say whether it is disease causing or not. The next step is the functional assay, which can put the ultimate stamp that whether this is a disease causing variant or not. So you could use different assays like the cell based assay, like the enzyme assays using site directed mutagenesis, Western blot, subcellular localization, real time PCR, luciferase assay for transcription factors, or you could use tissue based studies like induced pluripotent stem cells, or you could use model organisms like Drosophila, zebrafish, and mouse. All these studies are basically but to be done in a research setup and hence are not relevant to a clinical setup. So in a clinical paradigm, typically what you can use is either enzyme assay for lysosomal enzymes or some hormone assays depending on the phenotype, or you could use something called as a reverse phenotyping. That means once you find the variant and you know what disease is caused by that gene where the variant is present, you go back to the patient and use investigations like either biochemical or X-rays, MRI, CT, or clinical evaluation for dysmorphic features and see whether now the phenotype matches the variant that you have identified. So this is the reverse phenotyping that you need to take up once you identify a variant. So these are some examples of reverse phenotyping, like if the variant is found in a Hunter syndrome gene, then you will use the enzyme assay for Hunter syndrome or in the white cells or fibroblasts to confirm that this variant is disease causing or not. Similarly, in a patient who has features like limb girdle muscular dystrophy and you find variant in CAPN3 gene, which is known to cause calpainopathy, then you can do a muscle biopsy and do immunoblot studies to see whether it is really calpainopathy or not. Similarly, for a hereditary neuropathy, you could use nerve conduction latencies to prove that it is the same disease or not. So in this way, you can use different kind of clinical tests available to ultimately match the phenotype with the variant that you have found. So till now what we have done is just like a detective does different evidence collection from different sites. So we have done the evidence collection, but now the detective will present this evidence to the court and the judge will make the judgment whether it is criminal or not. But here we only have to be the judge, that based on all this evidence, now we have to decide whether it is a disease causing variant or not. So that judgment is going to be based on certain guidelines, and that is why the American College of Medical Genetics and Genomics gave these guidelines in 2015 about various criteria to be used to ultimately classify each and every variant that you get into benign, likely benign, variant of uncertain significance, likely pathogenic, and pathogenic. So every variant that you identify in any sequencing has to be put into one of these types. So different criteria are used, but if you see at the criteria, these are the same criteria that we have seen just now while collecting the evidence. So it uses the population data, the frequency in the population, it uses the computational prediction, it uses the functional data, the Mendelian segregation data, the allelic data, whether it is in cis or trans, which again is by Mendelian segregation, and then other databases like ClinVar or HGMD. So whatever we have discussed till now, the same evidences are used by the guidelines, and based on certain criteria, you will classify them into one of these groups. Now, this is a very complicated kind of classification, and hence again it becomes very difficult to humanly do it, and that's why we have tools available who make use of this for all particular criteria and ultimately give us the final classification for the variant. So ultimately we want to classify the variant into one of these, and a very useful tool for doing this is something called as Varsum. There might be many more available, but I'm just showing you this one as an example. It's available on this website. It's a free tool where you can put in your variant, the ultimate variant that you have identified into any of these formats. And once you put in, you will get a prediction as well as the basis for that prediction. So that is very important. No tool is going to be so expert that it is going to directly give you the answer. Every tool is going to help you. It is ultimately you who are going to be the judge, or you who are going to decide whether it is disease causing or not. So let us see some examples here. So when we put in this particular variant here in this gene, it is predicting as pathogenic. Now we cannot take this just on the face value that Varsum is predicting as pathogenic, so we say it is pathogenic. We have to see on what basis it is saying as pathogenic. So the basis are given here in the same ACMG criteria. So it is pathogenic because it is a null variant, so it is a nonsense variant. It is not present in the population databases, and prediction software are predicting it to be disease causing. So based on all three criteria, now we can classify this as pathogenic. Similarly, this particular variant in this ARSA gene, it is classified as likely pathogenic. And why is it? So this variant is not found in the population databases, but this is a missense variant. The earlier variant was a nonsense variant. So a missense variant is always difficult to classify. So here absence from the population and prediction still does not make it pathogenic because similar variant has not been described in any patient before. However, an alternative variant at the same nucleotide has been described as likely pathogenic earlier. So based on these three criteria, now we classify this as likely pathogenic. However, once some reports are available in this particular phenotype with the same variant, this will get reclassified as pathogenic. So it is very important for us to keep in mind that all these variants can get reclassified as time goes on. As more and more evidence is available, every variant can get reclassified into either towards the benign or towards pathogenic. If you look at this variant here now in this particular gene, so this variant is classified as variant of uncertain significance. So why is it? So this is a null variant. This is a stop codon variant. So that is why I said you should not jump to say that every stop codon variant is pathogenic, because although it is a stop codon variant, ClinVar has classified it as benign, and one prediction software is classifying it as pathogenic. So there is no enough evidence for it. There is conflicting evidence for us, and hence we are classifying this as variant of uncertain significance and not as pathogenic. Now looking at this particular variant here, we can see that this variant is not found in any of the population databases, but it is predicted as benign by the software and it is a synonymous variant. So now we will think, if it is a benign variant, it's a synonymous variant predicted as benign, then why are we saying it as likely benign? So we are saying it likely benign because it is not present in the population databases. But this may be because that particular ethnicity population might not have been studied well. So in future, if this population gets studied well, this may get classified as benign if it is found in normal population. So that's why it remains as likely benign here. Whereas this variant now here is classified as benign because it has high frequency in normal population, and it is classified as benign by the prediction software. So based on these two evidences, we can safely classify this variant as benign. So in this way, every variant can be classified, and tools like Varsam help us to do what was done earlier. But blindly we should not believe on these tools. We should be able to understand what explanations are given here, and based on that we need to make our own decision whether it is disease causing or not. So let us see one example of how we will use all these evidences. So like this is a variant found in GUS-B gene in a patient with mucopolysaccharidosis type 7. It's a missense variant, homozygous, and it is classified as likely pathogenic. So why it got classified as likely pathogenic? So first we go by the evidence that it is a homozygous missense variant in this exon and in this gene, and it results from alanine to valine change. Okay, so this is the location, type, and effect kind of a evidence. Then the next evidence is that this variant is not reported in public databases like 1000 Genome, Exact, Exome Variant Server. So this is the normal population database kind of evidence. Then this variant has not been reported in other patients of MPS 7. So this is the known variant database evidence. And then the in silico prediction software are predicting it as disease causing, and CAD score is 24, which is more than 10. So this is the in silico pathogenicity prediction based evidence, and the clinical features of the patient are matching with the phenotype which is expected in mucopolysaccharidosis type 7. So this is the genotype phenotype evidence. Based on all this evidence, we can now safely say that this is the variant which is responsible for this particular phenotype. Now, out of all these variants, it is very important to remember that genotype phenotype match, inheritance patterns, these are much more important compared to all these evidences. So if the variant is satisfying all these evidences, but the genotype and phenotype are not matching, then again we should not label this variant as the disease causing variant. We should always remember that the disease is caused by the variant, and these are all the evidences that we are gathering. So just because the evidence is present, it does not become the disease causing variant. The phenotype and the genotype always have to match for us to label that this variant is the cause of disease in this particular individual. Another very useful tool that I would like all of you to use is something called as Franklin. It is by a company called Genomes. This is again a free tool available where you can register, and then you can upload your VCF file, and then it will help you again to analyze the VCF file and give you the same filtering steps that we discussed. So based on the population frequency, based on the effect, the variant type, you can use different classifications to classify the variants that are available, and then it will give you each variant wise the information. So if someone is interested in doing the classification of the variant themselves, then this is a very useful and an important tool which all of you again can make use of if you want to analyze the VCF data. So what to do when a variant of uncertain significance is reported? This is a very important question which clinicians face. So one thing what you could do is that, because you have today attended this lecture, you can try and interpret the variant yourself. This is very important. Sometimes the lab might have missed something. So it is always good that if you give it a try to do the variant interpretation yourself. Secondly, try and check if the clinical features are matching with the phenotype. This is what is we called as the reverse phenotyping. Mendelian segregation, if it has not been done, very important to be done. Check if any functional assay is available for that gene and get the functional assay done. Very importantly, if you do not know, then do not reclassify it as pathogenic or something. You can tell the patient to follow up periodically, and you can check for reclassification of the variant. We have seen this many times happening, that some exomes which were done in maybe 2017 or 18 were reported as normal, or some VOUS is reported, and when we analyze the same exome now, we find that a disease causing mutation is present because gene was very recently identified. So very important to keep in mind that do not take any irreversible action like prenatal diagnosis, therapy, or genetic counseling based on a VOUS. It's always better to wait and see if the variant can get reclassified, and then only take up these kind of actions. So in summary, a proper interpretation of variant is very, very important compared to finding the variant, and knowledge is changing at fast pace, and hence clinicians as well as laboratory scientists have to be aware of the recent information, and we have to keep in mind the policies of variant interpretation, and so that we don't make any mistakes in diagnosis and counseling. And clinical correlation of variant and reverse phenotyping is very, very important in this era of huge genomic assays. So again, one more information that I would like to give you: if you are interested in medical genetics, please visit this website iimg.in of the Society for Indian Academy of Medical Genetics, where a lot of information is available regarding various genetic diseases, as well as pamphlets for patients, as well as 3-monthly journal that comes out, and so it can be helpful for you to be in touch with happenings in medical genetics. So I hope that all of you are now capable enough to try and do the variant interpretation yourself. A lot of material is already available through different websites or different tools, and we just need to be knowing the principles behind this so that we can make a correct interpretation. So thank you all of you, and thank you Premas for giving me this opportunity.
Thank you, Dr. Dalal, for insightful discussions. This forum is now open for question and answer session. So I do see some questions here in chat box, and some of you are coming to me directly. So I'll take up those questions one by one. So one of these common questions what we get generally in any of these variant interpretation and discussion and interpretation discussions, that how should we decide whether we should go for whole genome sequencing or whole exome sequencing?
Yeah, so that is same like deciding whether you want to buy a Maruti car or a Mercedes Benz. Yeah, so whole genome is the best, as we all know, because it will give everything. But again, the analysis is difficult and it is costly. But however, if you are able to do a whole genome sequencing, that would be the ideal test, because if you extract the exome out of a whole genome, you are going to get a uniform coverage of all exons, and hence whole genome would be a very useful test. But because of cost, we still prefer to do a exome sequence in the clinic as of now. But as I said, in near future, we are hoping that everyone would be doing a whole genome sequencing as the cost comes down.
Okay. And Dr. Dalal, where are we heading in rare disease diagnosis? As you know, day by day, new variants are getting added, and with respect to increased population. So where are we going to? I mean, how do we make a sense out of it? Like when we are getting more and more variants every day.
Yeah, so that is what. So with more and more variants, more and more confusion also comes up. So, and it is very important in clinical practice. The first principle is that do no harm. So our variant, a wrong variant interpretation should not do harm to the patient. So it is very, very important for every clinician to keep in mind that even there is doubt, do not label that variant as disease causing in that patient, so that some decision is taken regarding treatment or prenatal diagnosis. We should always try to be patient and make sure that this variant is really the disease causing variant in that particular disease. Enough evidence is available, and then only label that cause of that particular disease. Otherwise, it becomes very difficult to justify your actions after you have labeled that particular disease.
Right. And next question, what I can take up from chat box is, based on what criteria can we filter the annotated exome sequencing data for narrowing down to few clinically relevant or disease causing variants?
Yeah, that was the whole thing that we discussed today, that these are the criteria. As we said, first is the we would like to see the first criteria that you would like to see is that whether this variant has been reported earlier in a similar phenotype, which you could either go to ClinVar or PubMed, and if you find a particular article where same variant is reported and same phenotype is there, then you are going to report that this is a pathogenic variant. But if it is not reported, then you will go systematically through looking at the population frequency, looking at the predictions of the various software, as well as Mendelian segregation, and if any functional assay is available. And based on all these evidences and based on the ACMG criteria, ultimately you will label that variant as pathogenic or VOU.
And what would be the alternative if the mutation is in pseudogene or a region with high GC content, or if some epigenetic factors are involved with a particular disease?
Yeah, so these are, as I said, these are all the disadvantages of any sequencing. That pseudogene is a major problem. You might have seen in all exome reports, they will write that genes in the pseudogene will not, the variants will not be detected in an exome, because pseudogene can cause problem both in Sanger sequencing as well as next generation sequencing, because the reads will map to both gene and the pseudogene, and hence it is difficult to find variants in the genes where pseudogenes are known, like typically in Gaucher disease or in spinal muscular atrophy. And there you have to use other tests like either a long PCR or a allele specific PCR to identify the variant. Similarly, GC content is again a problem, and that I think slowly it is getting worked upon by different companies, that you get a better sequencing even in the GC rich regions. And epigenetic factors again is a problem, which again you will not get information in the exome sequencing or even a whole genome sequencing. And there, that is why I said a normal exome or a normal genome does not rule out a genetic disease. You will have to look for other mechanisms to look based on the phenotype.
Okay. And if a variant is reported as a VUS on ClinVar database, but in silico tools predicting it as a benign variant, and it's matching to the clinical phenotype of proband, kindly suggest whether it can be reported or not.
Yeah, so that's what you have to make use of all the criteria. So like you, as you are saying, in silico tools are predicting as benign, and it is classified as VOUS. So there you will have to see in ClinVar what criteria was used to say it as VOUS. Maybe it was not found in the population, and that's why they classified it as VOUS. So it will all depend on each variant. Difficult to say like this, but if you have enough information based on whatever we discussed today, you can now go and try to do it. And there are tools like Varsam available, which are there to make your job easy. But without Varsam also, you can just systematically go through each of those steps and ultimately make your own decision. As I said, here every person is a judge in itself. So what you interpret may not be same as what others interpret, but what you interpret will be correct only if you follow all the guidelines that are being laid down. If you put in your own personal bias into it, then it will go to a wrong interpretation. So I think be very objective, try to use the evidence in a very objective way, and then let the variant remain as VOUS. Don't try to classify each and every variant that you get. It's not your responsibility to classify every variant. There's no shame in saying that I'm not able to classify this variant at this time. Maybe next year you will be able to classify it.
Okay. And another interesting question is how to decide on the coverage required or I mean, how many GB data is required to target while sequencing?
Yeah, so that we have certain like in NGS, when we want to call a particular variant, we need at least 20x coverage for a particular variant to call it confidently as a variant. Now to get 20x, typically what people feel is that 80 to 100x depth should be there for a exome, because exome is a very it's capture based. So you're not sure if you go for a 20x average coverage, in some places it will be 1x, some place it will be 50x. So that's why if you go for an average coverage of 80 to 100x, then you get roughly 20x throughout your exome. But the same thing if you are going for a genome, because genome is a uniform kind of coverage, there typically we will go for around a 30x coverage to get enough data to interpret the results. So this is a ballpark figure that people use.
So, sir, we have been doing a lot of sequencing, and there are a lot many sequencing projects there in different institutions. So, have we reached the satisfactory stage in the clinical utility of exome sequencing? I know it's a never-ending thing. I mean, these things will be going on and
But
on.
see, even this all these tests are being available again in major cities, and also there is a huge population which still needs this sequencing, and there is a huge need for this kind of sequencing, because as we know, rare diseases almost 350 million are there throughout the world, and of which
Correct.
about 70 million are in India itself. Now, I don't think we are able reaching all these 70 million people. So many of these rare disease patients, either due to cost problems or due to unavailability of the test, they still remain undiagnosed for a long time. So I think a lot needs to be done in this area, and hopefully as more and more labs start doing sequencing, and I hope companies like Illumina and Premas bring the cost down, we are looking at a lot more sequencing to be done in future.
Great. Thanks, Dr. Dalal. I would like to wind up here as I see no more questions in the chat box. So I'll thank you for an insightful discussions, and we are looking forward to having you again for any such event in future. Thank you very much for being part of this webinar, sir. And I also like to thank the audience for their enthusiasm and proactiveness in today's session. I saw quite interesting questions in the chat box for Dr. Dalal. And for whosoever the audience, please reach out to us with ideas and suggestions. We would like to be part of any such program and love to extend the support required from all the sides. So thank you again for your presence. Please take care of yourself and have a great rest of the day. Bye-bye. Thank you.
Thank you.